Good morning, everyone. Good afternoon and good evening to everyone who is joining us today, and welcome to today's webinar. Today's talk is entitled Risk-Based Fraud Detection, How Centralized Monitoring Can Boost Data Quality. My name is Kelsey Brown, and I am the Director of Medical Writing Solutions at TransPerfect Life Sciences in London, and it's my absolute pleasure today to be your moderator for today's session. Today's event will run for approximately 60 minutes, and that includes time for Q&A with the speakers at the very end. You can go down to the next slide. All right, so just as a reminder, this session is designed to be interactive, and it works best when you're involved. So please feel free at any time to submit questions and comments for our speakers by using that questions chat box in your control panel, and we'll answer your questions during the Q&A session. And if you require any assistance at all, please contact me at any time by sending me a message using your control panel. At this time, everyone involved is on listen-only mode if you're part of the participant group. And just to please note, this session will be recorded, so it'll be made available to you on the IDDI website for viewing about 24 hours after we wrap up the webinar. You can go to the next slide. So this event is proudly hosted by the International Drug Development Institute, IDDI. IDDI is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI is headquartered near Brussels, Belgium, with the U.S. Center of Operations in Raleigh, North Carolina. Next slide. And today's speakers will be introduced by Mark Busey, Chief Scientific Officer at IDDI and founder of CluePoints. So I'll hand it over to you, Mark.
Thank you, Kelsey, and good afternoon or good morning, everybody. My name is Mark Boyce, and I'm going to introduce the two speakers, Laura Trotta, who's the Vice President of Research at CluePoints, and Sylviane de Viron, who's the Data and Knowledge Manager at CluePoints as well. So the agenda of our meeting is as follows. We will proceed in 3 steps. I will begin to introduce central statistical monitoring, and then I will pass the floor to Laura, who will actually describe the CluePoints monitoring platform. And finally, Sylviane de Viron will show a really interesting case study where a case of fraud was detected in an ESPS2 trial using the CluePoints engine. So let me get started with central statistical monitoring, and what do we mean by that? Well, this whole adventure started more than 20 years ago now, when we published a paper in Statistics in Medicine on the role of biostatistics in the prevention, detection, and treatment of fraud in clinical trials. At that time, this was, kind of science fiction, because there was no software at the time that was, in fact, available to implement the ideas that we described in this paper. And so it took about 10 years before we actually developed the software implementing the methods that we had advocated in the first paper, and that we had an engine, if you like, a software engine that would actually detect problems in the data, not just fraud, as Sylviane will show, but also all kinds of problems, whereby you can detect actually problems in one center by comparing that center with all the other centers in the trial. So that's the main idea in the CluePoints engine, is to look at each center at a time and compare whether that center, in a sense, fits in with the distribution of the data with all the other centers or not. And so this is, in fact, a very powerful approach, because no data are left unanalyzed. The engine really looks at all the data, whether they be clinical data or operational data. Everything is looked at. And so the engine really has a lot of power, as we will see later, to detect problems. And the problems can be of any nature. Fraud, of course, is one big problem, but there are many other problems: sloppiness, miscalibration of equipment, and so on and so forth, as we will discuss later. And so the idea, again, is to take one center that participates to a clinical trial, and then for all the variables collected in that center, and for all the tests that we can perform on these variables, we will actually do a lot of tests. And the test can bear on the mean of a continuous variable distribution or the variance of that distribution, on the proportion of the successes or failures, etc., etc. There are many, many tests that can be applied to the variables. So suppose, for example, we have a variable called weight here, which is the weight of the patient. We can actually look at the mean weight in a center, the variance of the weight in a center, outlying patients in a center, missing data for weight in a center, and compare one center, here called the center X, with all the other centers in the trial. And we would expect, just because the trial is of course standardized and by a protocol, and all the centers have to follow the same protocol, we would expect that the data in center X are consistent, broadly speaking, with the data in all the other centers. And so this tool or the software will really detect any inconsistency between the variables collected at center X and the variables collected, or the values of the variables collected at center X versus the values of the variables collected in all the other centers in the trial. So what we do again is to look at all the variables and run a number of tests. Suppose that, for example, we do a trial that has 400 patient-related variables. So we collect 400 variables for each patient, and we can run, let's say, 5 tests per variable, and we have 50 different sites or 50 centers in our trial. Well, you multiply 400 by 5 by 50, and that's 100,000 p-values. So we generate a lot of p-values, even in a relatively modestly sized trial, simply because we do a lot of tests. And the sorts of tests that we do are listed on this slide. The smart engine will look at reporting rates, missing values, event rates, proportions, transitions, and so on and so forth. So lots of statistical tests depending on the type of the variable that we look at. So we end up with 100,000 p-values, and obviously no one knows what to do with 100,000 p-values. That's far too many p-values for anyone to look at. So what we do is we actually store these p-values in a table. And this table has as many rows as centers and as many columns as tests by variable. So for the weight, for example, remember we had looked at the weight, we can have a test on the mean. Is there a difference between the mean weight in center X versus all the others? There is a test on the between patient variability. There is a test on the within patient variability if the weight is repeatedly measured over time, and so on. So we have lots of different tests for all these variables that we've collected. And that table, therefore, will have about 100,000 cells, as I just argued. And how do we actually summarize all these tests? Well, we look at one center, which is one row in this table. And what we do is we actually summarize all the p-values. See, all these numbers in these circled cells here are p-values. And we actually summarize these p-values by a single statistic. And we don't need to go into the details of how we do that, but we calculate what we call a data inconsistency score, which is really a summary of all the p-values for a specific center. And so each center, at the end of the analysis, each center will have a DIS, a data inconsistency score, which summarizes, in a sense, how extremely different from all the other centers this particular center we look at is. And so that will allow us to rank the centers by the most discrepant to the least discrepant. And of course, we will focus our attention on the very discrepant sites, as we will see later in the example. But before we go to the example, I would like Laura to give more details on the CluePoints monitoring platform. I've just shown the basic principle of the data quality assessment in CluePoints, but there are other features in the monitoring platform that I think are worth describing. Laura? Describing. Laura? Describing. Laura? Describing.
So as Mark mentioned, within CluePoints, we've been developing a web platform to monitor data and to look for risk in clinical trials. And it's really based on these ideas that was presented by Mark of using central statistical monitoring to interrogate all the data collected during study conduct to look for any potential risk within study conduct as well. And go to the next slide. And so there are many things that can go wrong in a trial. Of course, we have the basic, missing the basic, that side, some safety issues that are putting the patients at risk, miscalibration of equipment, failure to enroll patients at the site. Patients may be on hold at multiple sites or receiving potentially several times the treatment, some GCP non-compliance, or even data tampering or fraud. So if we go to the next slide, 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 some operational data to look for any potential risk in the study conduct. And so we're going to inject CRF data, so electronic case report form data, EDC data, but also lab, ECG, ePro, ECOA, and some CTMS data. And in what we call our data quality assessment, we're going to interrogate all data in a completely unsupervised way, meaning that we're going to interrogate all variables together and generate these huge matrix of p-value and compute this data inconsistency score. And in that specific view, this is a view of web platform that is actually displaying the data inconsistency score analysis for a clinical trial. This is what we call the bubble plot. On the x-axis represents the number of patients by site, while the y-axis is representing this data inconsistency score that was introduced previously by Mark. The size, each bubble here, it's a site, and the size of the bubble is also the number of, represents the number of patients enrolled at the site. So the higher the DIS, the most atypical the site is with respect to all the other sites within the trial. And this is really taking into account all the statistical tests that have been performed for these specific sites, considering all the variables included in the trial. Within CluePoints monitoring platform, when a data analyst, either a CluePoints data analyst or a sponsor data analyst or CRO data analyst will review this bubble plot, the data analyst will have the possibility to dig into the results and start exploring why we have such an atypical site with respect to the other sites. And typically we'll access a screen like this screen where each triangle square is representative of the results of one single individual test. So on the x-axis here, we have the score, which is in fact a log transformation of a p-value. So the smallest the p-value, the higher the score, the most atypical is the statistical results. On the y-axis, we have all the results of all the statistical tests that are grouped by domain. So we have, for example, in that specific example, laboratory test results, questionnaire data, vital signs, ECG, subject visits, but we have can have many more data sets and domains within the trial. And the data analyst will also have the opportunity to further dig into the results of one statistical test and look at some summary about the test and access the patient level data. In this specific example, we see that we have a significant score for a test on propagation of data in the vital signs data sets for the weight measurements. Basically, it means that for this specific site, the staff at sites has been measuring the weights, but it seems that there is a high number of propagated value across successive visits and across different patients within the sites, pointing towards some potential anomaly at that site. So if we go to the next slide, the data analyst might decide to create a signal to report these potential issues to the study team. Within CluePoints monitoring platform, we have a signal tracker that is going to make possible the fact that you report all the data anomalies and document all these anomalies to the study team. So the system is not including a signal tracker, but also an action tracker so that the study team can follow up on those risks and take some mitigation actions to maybe mitigate the risk during study conduct as well. If we go to the next slide. So I've been really focusing here on what we call our data quality assessment. And this data quality assessment is really aligned with what Mark was presenting before of analyzing any potential variable in the trial. Indeed, and as Sylvian is going to show in the last part of the presentations, we may not really know in advance where the issue might arise, and we may want to explore all potential variables to look for any potential anomaly at the site. However, sometimes there are some specific variables where you already know that you would like to focus on these specific variables because they are key risk indicators. Within CluePoints, we also develop key risk indicators that allow you to perform a targeted analysis by focusing on these specific key risk indicators, both for operational and clinical data. So what is specific to CluePoints is that usually these key risk indicators are based on what we call absolute value, but within CluePoints, we have been using the results of the statistical analysis to complement these KRIs with relative score as well. So here I have an example of an operational KRI. So they have from operational data, not clinical data, which is the visit to entry cycle time. So basically the average cycle times from patient visits to EDC entry. And so in a specific setting, the user, if he's focusing on the absolute threshold KRI, may decide to fix a specific threshold for which an action should be taken if at that specific site, the number of days is exceeding a specific limit. In that specific example, the user decided to select 15 days as the number of days needed to take an action. A different way of seeing this problem is I would like to raise a signals whenever my site is starting to be atypical with respect to all the other sites that I have within my study. And in that specific case, instead of using this absolute value threshold, the user will use a relative score derived from the statistics. So what you see here with this score is in fact the results of one single statistical test. So one cell in this big matrix of p-value. The score is once again a log transformation of the p-value. So a score of 1.3 would correspond to a p-value of 0.05. So if basically the KRI is exceeding the threshold, that means that statistically the site is atypical with respect to all the other sites. And in some specific setting, that might be useful to use a relative score. Maybe for operational data, you have a good idea of what should be the operational threshold, like these 15 days. But for other key risk indicators, it might be difficult at the beginning of the study to set up a threshold for the analysis. And in that case, it's meaningful to rely on a relative score to assess if some specific sites needs to be further investigated. We can go to the next slide. Here are some examples of KRIs that we usually, that are part of the, I would say, standard list of KRIs provided by CluePoints. Of course, we have some KRIs focusing on safety, like adverse event rates or serious adverse event rates, but also on data quality, like the the auto query rates of this visit to entry cycle time, or some other data quality like misassessment rate, so how much of the assessments were missed during study conduct, and maybe also some KRIs linked to enrollment, like screen failure rates or early termination rate as well. I've been mentioning that we computed these data quality assessments and these key risk indicators by site, but the system is in fact offering you to do the same by patient or by country or by even, I would say, state, if you would like to look for any other dimension of risk so that you would detect not only atypical sites, but atypical patients and countries as well. If we go to the next slide. So within this presentation, I focus on two specific products within our CluePoints monitoring platform, the data quality assessments and the key risk indicators. But we have 6 products within CluePoints that are there to monitor and document risk within the full study cycle, starting from the risk assessments towards database log and submissions to agencies. And we can go to the next slide. So, as Mark said, journey of CluePoints started more than 20 years ago with these publications in the role of statistics in the prevention of fraud in clinical trials. But Within the years, we have been analyzing more and more studies. We have detected more and more risk. We have documented those risks within our signal tracker and signal management systems. And so as we've been analyzing more and more trials, we are now able to use those metadata to not only do a supervised analysis based on statistical methods, but to try to do supervised learning as well and use machine learning methods to complement our solution. More recently, over the last 3 years, CluePoints really switched from pure machine learning, I would say, to deep learning and natural language processing. And this switch has really been triggered by the pharma industry itself, because the pharma industry is recognizing the important role that those new techniques will play in the future of clinical trials. We have been involved in several hackathons with top 5 pharma companies. Last year, we released our first version of first NLP solutions within the system that is basically screening all those signal documentations real time to further categorize those signals and anomalies. And we are trying to extend the solutions not only for looking at risk-based monitoring, but also for data management and medical review activities. We can go to the next slide. We are very happy that we've been recognized as the Clinical Advance of the Year for the work that we did together with Pfizer and BioNTech in supporting the COVID-19 vaccine trial. And as of today, I think within CluePoints, we have analyzed more than 900 studies. I think we are reaching the 1,000 studies at this stage. We've performed more than 400 data quality assessments, identified 100,000 risk signals and 20,000 resolution actions. So I think I'm going to let now Sylviane present this very specific use case, which is the ESPS2 trial, which is really about a very famous case of fraud within clinical trial.
Thank you, Laura. I will present you this case that we published really recently. This paper is about ESPS2. There was a study from Boehringer Ingelheim, and we published this paper in collaboration with them to show really to see if CluePoints was able to detect the fraud in that specific clinical trial. In ESPS2, this trial was specifically looking at the prevention of stroke or death in patients with pre-existing ischemic cerebrovascular disease. It was a phase 3 multi-center randomized double-blind trial. This study was a very big study with more than 7,000 patients, 60 centers, and 13 countries. And we compared treatments that were acetylsalicylic acid with dipyridamole versus placebo. For those who know stroke disease, you would know that those are no more the standard of care. And you will see that this study was run in the 1990s. It's a very old study, meaning that the results we provide here with this analysis are not changing anything into the standard of care of the stroke disease, because those are very, very old treatments. Why we selected specifically this study? It's one of the rare studies for which we know and there is a public knowledge of fraud in a study. In this study, indeed, in the Center 2013 that enrolled 438 patients, we found, or Boehringer Ingelheim found, serious inconsistencies in the case report form. The treatment concentration that they collected was totally implausible, and due to those results, the trial steering committee decided to exclude the data of the center when making the final analysis. We received those data, and the objective for CluePoints was really first, are we able to identify the center with known fraud? Just a side note, but an important one, the clinical team or the data analyst team that worked on this trial was not aware of the nature and the scope of the issues, so that we were absolutely not biased when making the setup. We really make the setup of the study as we would have done for any other study. And then the second question, if we are able to flag this center with known fraud, by when are we able to flag it? How early are we able to flag this, this specific center? And to do that, what we did is really like rerun the analysis by always reducing 5% of data volume and trying to go as with the lower data volume as possible. Yeah. Here in this paper, we only focused on data quality assessment, but when we did the analysis, we also ran KRIs and other types of analysis for our knowledge. But really, like the purpose of this analysis and of the today's presentation is more to show the data quality assessment, which is really unique of CluePoints. This is really like a unique solution we have. As Laura and Mark already said, really like the goal of this data quality assessment is to identify atypical centers through finding anomalies, discrepancies that we would not be able to flag via on-site monitoring. We know that making manual check, it's good, we can spot some issues, but we will not be able to spot all types of issues. The type of data discrepancies that we are able to flag using our data quality assessment is inappropriate training, a poor understanding of the protocol or the completion guidelines, sloppiness in the collection of the data, and in some cases, fabrication of data or real fraud. The data that we received from Boehringer Ingelheim was really like all the ESPS2 data. It was both clinical data and all the laboratory data. And based on that, as it was also mentioned before, we decided we applied tests on all collected data, on all the data available in the ECRF, and at the end we reached 838 statistical tests in each of the 60 centers of that specific trial. If you look at the table, you can see that we have really like diverse domains where we apply tests. It can be adverse events, it can be clinical events, concomitant disease, and so on. The tests that are most frequently applied are data reporting. It's mainly looking at missing values. And in this case, in this very specific study, we had a lot of categorical variables. It's a very old studies where they collected a lot of binary or factorial variables. We have also some a fair number of continuous variables there. And as you can see, it's mainly about labs and clinical events data. Can go to the next slide. This is the results of our analysis. As you can see, we have here the bubble plot, as Laura already presented. On the y-axis, we have the data inconsistency score. This is really like the summary of all the p-values that we retrieve for each of the center. And a data inconsistency score of 0 means that the center is not atypical at all. If it's closer to 10, we have something very atypical. You can see that we have 5 bubbles that are in magenta. Those ones are the significant centers in our trial. And our center 2013 is the rank 2 center, the second most atypical center of the trial. It means that our center with known fraud is already really like atypical with a DIS of 4.14, but we have even one center which is even more atypical, and that's our center 8. An important other note for the center 2013 is that it's, as you can see, the bubble is really large. It's the 4th enrolling center in the whole trial, meaning that, as I told you, Boehringer Ingelheim had to exclude this center. It's already a lot of patients that they had to exclude from their analysis. And for which reason was this center specifically outlying, the center B, center 2030? In fact, in this center, they only reported serious adverse event. No other type of adverse events were reported, meaning no non-serious adverse event, no bleeding. And just to remind you, we're here in a stroke prevention of stroke trial. The patients receive anticoagulants, meaning that if in 438 patients there's no bleeding, it's really suspicious. But those very first findings are stuff that we could also find using KRIs. That's not really like specific to data quality assessment. While the next one, a low between patient and within patient variability for lab results and vital signs. This is something really like that we would not be able to flag via any other manner than using a data quality assessment. And we know that if someone is sloppy or want to manipulate data, being close to the average, having a correct mean is something that we can easily manipulate. But for human to have the right variability is something very difficult. And having such kind of findings should provide already some insights that there might be something wrong in that center. We had also other types of findings like atypical proportion, large number of missing values, and so on and so on. Meaning that if we were in an ongoing trial with such kind of results, for sure we would need to look more in depth into those results. We would for sure need to go into the center, retrain most likely the center so that they can improve their data in the run of the trial. Center B was the second most outlying center. But as I told you, we have also the center A, which is even more outlying. But the story there is a bit different. If we look at that center, we can see that it's a very old population. At baseline, when starting the trial, this population had more concomitant disease. They had more abnormal assessment. And during the follow-up, during the conduct of the trial, they had more severe bleeding, more abnormal stroke assessment, neurological examination, lab results. It means here that we are more with a very atypical population. All those signals, all those things that we flagged are not really like related to something suspicious about data fraud, data loss. It's really like we have here an atypical population. And if we were in a prospective study, in an ongoing study, most likely by detecting this center, we would have a closer look at this specific center. Most likely medical reviewer, medical people will make sure to follow up this center for safety reasons and so on, because we know they might be at higher risk. You can go to the next slide. Here, it's just to present you a bit the difference of what we found using our analysis, the CluePoints platform in Center 2013 versus what the sponsor, Boehringer Ingelheim, was able to find in the past. For adverse events, they found that there was a very low incidence of adverse event. We found that there were no non-serious adverse events reported, too few serious adverse events, no patient with vascular or bleeding events. For the vital signs, they flagged that there was very low variability in blood pressure reading. We were able to confirm that, or to have the same results, very low variability between and within patient at the center for pulse rate and systolic blood pressure. For the study compliance, they were able to flag a very low variability in the counts of return study drug capsules. We were able to find the same kind of findings. Patients were highly compliant with the study drug regimen. And regarding the clinical lab data, the sponsor didn't publicly reported anything, but we were able to flag very low variability between and within patient for numerous lab measurements. Now we flag that here, without any doubt, we are able to with all the data, when we are at the end of the trial, we were able to flag this center, the center with known fraud. The next question is, how early are we able to flag this center? And when we did the analysis, we really reduced the data volume of Center 2013 and all the trial by 5%, and we were able to go down up to 25% of data volume. And with that, we still had a significant p-value of DIS of 1.73. We had still 205 patients, meaning a bit less than 50% of the patients. And at the very big, when we had all data, 100% of data volume, we had 48 significant tests. With 25% of the data volume, we still had 21 significant tests, meaning 44% of the tests that were still significant. It does not mean that before 25% of data volume, we would not have flagged this center. When we look specifically at the 20% of data volume, for example, the p-value is 0.07, meaning that's really borderline. And in that center, there were already 163 patients enrolled, meaning that it was already a very large center with a very borderline p-value. Most likely, when we see such kind of results, we are looking into the site just to get some insights and see if there might be some issues. But we have the data quality assessment. We have also other kinds of tools that could help us to flag the center. And for example, the KRI was already significant for the AE rate and SE rate, meaning that the 25% was the very first time we were able to flag the whole center as being atypical for the data quality assessment, but it's not that we would not have been able to already get some insights before. An important point is that CluePoints data quality assessment identified the fraudulent centers issues 39 months earlier than what the sponsor was able to do. And I think that's a very nice message here showing the power of our tool. Now with CluePoints experience, we already discussed about a lot about our experience, more than 900 studies. We can say that having a real case of fraud is something really rare, while having other types of data errors like sloppiness, lack of understanding, or training is something more really common. In most of the trial, there is at least something that's not well understood or some data entry issues. Usually, there will be always some stuff that we can improve in the quality of the data. The cost of central monitoring is much lower than what is human data check. Central monitoring, what you need to do is do the setup of the study once, and then refreshing the data and having updated analysis is something question of 1 hour, 2 hours, and you get each time new analysis and new insights about your data. While human data check, you need to send someone to the site, you need to look at the data manually. You can imagine that the cost is much higher, and the efficiency is much lower, because for human being able to flag, for example, the variability issue as we saw in our ESPS2 trial is something very difficult, because we have no real knowledge of the variability that's expected, while central monitoring is able to get this information. If we do not detect a center with an issue at the end of a trial, there is a high risk that we will not have enough power to prove our key study endpoints, which would be a very important issue for the sponsor. The second risk is that if we send the data to regulatory agencies, is that they are doing other types of analysis, some central monitoring analysis, and they may flag your center with an issue. In that case, there is a high risk of invalidation of your study. Then the last point is about if we do not detect the center with an issue early enough, as you can imagine, we have a high impact, really like high direct cost impact, because we need to pay the center, we need to supervise the center, everything like that is really like expensive, as you can imagine. And last but not least, if we know that a center or if there is a center not doing their job properly, there are also some safety risks for the patients. If they are not following up properly their patients, if they are not providing the treatment as they should, the patient is taking or may take more risk than expected. In conclusion, unsupervised statistical analysis is a powerful tool to detect fraud, as we have seen, but we could also find various types of risk during the conduct of a trial. We could detect like data errors, sloppiness. We could detect very different types of risk. And detecting those risks early in the trial is very important so that the study teams are able to take the correct action to correct their data so that at the end we have very good data and the best data possible.
Thank you very much, Sylviane and Laura. So this gets to the Q&A section of your presentation, and we're going to look at some of the comments we've received. I'm trying to look at the comments here, and I will need some help from the organizers of the webinar because I don't know how to display this these comments. So I will ask. Yeah.
Yeah, I don't think we actually have any questions yet. So if anyone from the audience would like to send in some questions, you can submit those in the control panel. I don't think we've gotten any questions yet so far.
Okay, well, but before we get the questions, I had a question myself for Sylviane, because she's very familiar with the ESPS2. So what you've shown is that there was a major case of fraud in this study, 438 patients. So that was a really large number of patients, but you did not tell us exactly what the fraud consisted of. What were these patients? I mean, these 438 patients, what was wrong with them in a sense?
In fact, when we discussed with the sponsor, what we discover or what we learn is that, in fact, those patients were not really enrolled in the trial. Those were really patients from the daily practice of the doctor. He just took data and then he took some blood and he used a bit of all those data to create a fake center, and that was really like the output of that specific center.
So would you conclude from this single example that it's actually very hard to invent plausible data?
I think this is clear that inventing data is really hard. It's clear that at least being able to reproduce the variability in the data is something really hard for a human. Maybe now, if someone is starting to reproduce the data with very sophisticated tool, they would be able, but a human, like just doing it manually, is very hard.
Okay, thank you. We have a question from the audience on the QTL, the quality tolerance limit. So I guess that's a question for Laura. And the question is, can you expand upon the inclusion of QTLs in your engine?
Yes, so I think we can go back to one of the slides.
Would you like me to go back?
Yes.
Okay, we can do that.
We show the 6 different products. Yeah, here it is. So as you mentioned, within the tool, we also have now the solutions offering our customers to also use and specify some specific quality tolerance limits as well. So as of today, the idea is for the systems using the tools to have a quick access to what we call the expected value of the trial. So basically, when we compute the statistical analysis, we compute what we call the observed value at the site, but also the expected value across all sites. And typically, a QTL would be a quantity computed overall across all sites for your trial. So it's a global quantity computed a quantity tolerance limit, so that they will be notified if suddenly by analyzing the data from all sites, the expected value is reaching the quality tolerance limits. Hope it's providing an answer to the question.
Well, it looks like it. Otherwise, the person can certainly follow up on that question. But I had a question on the quality tolerance limit, because for the key risk indicators, you made a difference between specifying absolute values for the key risk indicators versus looking at the score, which is really a statistical concept of whether a value is abnormal statistically. Do you have a similar mechanism for quality tolerance limits?
As of today in the platform, quality tolerance limits mainly remain on an absolute value. The team is working on new methods to also have assessments more like relative score as well. But we know that quality tolerance limits are specific in the way that a quality tolerance limit is going to be very specific to study indication, therapeutic area, and you have a lot of variability within clinical trial. So when we do a key risk indicators, we are comparing data across different sites, and we know that by design, the multi-center clinical trial is designed so that you have some variability, but that is going to be captured by the model that we're using for statistical analysis. With the quality tolerance limits, we have this extra complexity that is coming from the fact that you're going to observe very different QTLs depending on the study indications and therapeutic area, and taking that variations into the statistical methods is the main challenge.
Yeah, and I have a follow-up question on this issue, probably from the same person. And the question is, are thresholds fixed? So I guess for QTL, or do you use some version of statistical process control? So I guess that has to do with the dynamic progress of the trial over time and how you change your QTLs accordingly.
Yes. And that's a very good point, because basically in the next version of the platform, we've been implementing a version of where the not a fixed threshold anymore, but a threshold that would vary depending on the size of the data collected during the study, so that whenever you have the beginning of the study and the volume of data collected is low, if you define your QTL like a proportion or counts or relative measurements, you would allow the systems to have a higher limit to account for the natural variability that can happen due to the low sample size. And so the limit is not going to be a fixed threshold anymore, but a curve that is going to take into account the volume of data collected at the sites. So that is coming to the next version of the platform, and that's what we call the adaptive QTLs. And in that regard, it takes this variability into account, but this is still specific to one study at a time.
Okay. Okay, thank you for that. Now I have another question here, more on the risk-based monitoring in general. And the question is, can the risk-based monitoring be implemented within the platform? So I guess you touched upon that partly when you described the signals and the actions, I suppose.
So the question is, can we implement RBQM with the platform?
Can you implement RBM, risk-based monitoring, within the platform?
Sure. This is the goal of the platform. So it's a RBQM platform, risk-based quality management platform. So when we say we implement risk-based quality management, it goes from really like the risk assessment. So prior to the studies, where you review a protocol and will identify the risk. Then you create some mitigation actions. So mitigation actions can be going to create some key risk indicators to follow up on specific risk, or can be this data quality assessment. For example, every 3 months, I would like to run a data quality assessment to look at all the potential issues at my sites and risk at my sites. And then, of course, you're going to document the risk and follow up on the risk using the web platform and the signal management and action management tracker. And we also have an integration with a CTMS system, so that during study conduct, you can properly follow up on those risks and send people on sites, or take specific actions to correct those issues at site. So it really goes from, I would say, risk assessments to the full trial cycle. And this is this idea to implement some mitigation actions to look for any potential risk and implement corrective actions as well.
Okay. Well, thank you. I have a follow-up question, I think, from the previous person, I think, who was interested in the, I don't know who asked the question. I cannot see this on my screen. So all I see is the question, but this question seems to me related to the previous one about the QTLs and the adaptive QTLs. And the question is, when will this next version be released? I guess that's a tough one.
So it should be in 2 months, if I'm not wrong, but I always need to go back to product team to confirm, because we have within the research team, we are mainly focusing on research innovation, but the next version of the platform is going in 2 months. So
so is that the routine, that the platform is updated every 6 months? Is that how it goes?
Yes. We have 2 major releases a year.
Yeah. All right. I have a more general question about, centralized statistical monitoring, which you've talked about, both of you today, and on-site monitoring. And the question is, does centralized statistical monitoring allow you to reduce the on-site monitoring intensity?
So the question is this helping reducing the, I believe so, and what we've seen, especially with the COVID-19, is that in some specific situations that was, I mean, really helpful, because in the case of the COVID-19 crisis, in fact, most of the people could not go to site anymore. And in that specific context, we've seen that the platform turned to be very useful, and we've seen really a huge increase in the number of trials analyzed on the platform at that specific time, because people were looking for solutions to really look for a remote control of the remote assessments of data. And so in the context of the COVID-19 crisis, we've really seen this shift happening. When some people were still heavily relying on on-site monitoring, they turned to remote monitoring to control and check the quality of the data and processes and identify risks within the trial.
Okay. Another question on the site experience. So the question is, does the site experience and location, is that taken into account when the analysis is done within the platform?
Not directly. So the statistical methods are designed in such a way that they are not sensitive to variations, natural variations that might occur in multi-center trials due to geographical locations or any other specificity of the site. So we model within the statistical test this natural variability that can occur across sites, so that we do not flag a specific size because of an atypical geographic location. For instance, let's say that we have a specific population that is going to be modeled, but we do not put some a priori information regarding the locations and the location of the sites in terms of risk. So we do not set an a priori risk based on the location of the sites or the size of the sites. We have an approach which is completely agnostic of the location and size of the site to detect risk anomalies.
Now, if we go one step further, you said that the COVID had been a special, obviously a special era, because a lot of site visits were impossible. And so, of course, that has been a big push to for the centralized monitoring idea. But I have another question, which is, the COVID-19 also has promoted the idea of decentralized clinical trials, which are very popular these days. And so I wondered how CluePoints could be applied to DCTs, decentralized clinical trials, because you no longer have the notion of a site.
Yes. And it's a very broad question, of course. In the decentralized trials, you have, of course, different modes of decentralized trials. A fully decentralized trial would not involve any site anymore. It would just be remote patients or remote collection of data, and patients would not even go to sites anymore. But we also still, I mean, seeing some decentralized trials, but where we have like still patients going to sites, but they are more and more relying on these technology like ePro data or ePro data to collect patient some assessments remotely as well. So it's true that in the context of fully decentralized trials, the notion of site disappears. Within CluePoints monitoring platform as of today, as I was saying, the site is not the only dimension of risk. We are also analyzing patient and country as specific risk factors. So we would still be able to run in that specific context the analysis to detect atypical patients or atypical countries. But you also have maybe new dimensions of risk. For instance, if you have healthcare, somebody going to the site to visit the patients to help them in assessing and understanding how they are supposed to take measurements. Let's assume this person is not going to explain properly the way the data should be collected to the different patients, then it becomes a new dimension of risk. So we have new dimension of risk due to this decentralized nature of the trial. And CluePoints is looking for assessing and developing methods that would especially target those new dimensions of risk as well. Another interesting thing is really the fact that we have different modes of data collection. let's assume that we are, for example, what is the impact of collecting some data through some electronic device when before it was collected by going on site, and assessing the impact of this different modes of data collection is something we are also looking at right now.
Thank you. And I have a question for Sylviane now for a change. Sylviane, you described in your presentation the sorts of problems that were detected in ESPS2, but the question here is, can you give us an idea as to what types of errors are most commonly found in the CluePoints platform across all the trials that you've looked at already? That's a tough question because I don't know that you have these summary statistics, but
I don't have these summary statistics, but my feeling as previous data analyst is that we have a lot of data entry delay. I think that's part of the things we flagged. Data errors, really, like either the labs were not in the right units of measurements, and we flag those results so that we are able to correct those results. We were able to flag miscalibration of technical, like a blood pressure tool, but that's less frequent, as you can imagine. It's not something really common.
And if I may ask a sort of sub-question, how frequently do you find real fraud, like in the ESPS2 study? Is that frequent, or because no one knows really what the incidence of fraud truly is, because it's underreported in many cases?
It's underreported, and I think even for CluePoints, even though we are working with data, we don't always have the truth from the sponsor, because we are not the one. We are not in all the meeting with the sponsors.
That's a good point. So there may be more fraud than you think, in a sense.
Yeah, and sometimes we don't exactly know what's really like the output of what.
And if an investigator, for example, doesn't have blood pressure at each visit and they just make up some numbers, is that fraud?
That's also a good question. What's the very real definition of fraud? Is just sloppiness? I think now with the ESPS2 trial, we have such a huge case of fraud that there I think the definition is quite clear. But really, like more sloppiness, like I don't want to take the blood pressure each time because is it a real fraud, or is it sloppiness, or is it?
Technically, it is.
Technically, it is. Then most likely we would detect more fraud if we work with.
If you knew the truth.
Yeah.
But that we will never know, of course. All right. I think we've run out of questions from the audience here. So, and we are not far from the top of the hour. So I think this is probably a very good time to finish the talk and to finish the presentation. So I'm going to hand back the control to Kelsey for the end of this talk. And thanks very much, Laura and Sylviane, again, for a very nice presentation. And now back to you, Kelsey.
Thank you, Mark. Thank you, everyone, for your questions today. We've got some really great questions. We've reached the end of today's session. So just a note, if you have any further questions, you can direct those to info@iddi.com. That's info@iddi.com. We thank everyone for participating in today's webinar. Just a quick note, you will be receiving a follow-up email from us with access to the recorded archive for this event, and you will receive a survey window that will be popping up on your screen after the webinar. So your participation is very much appreciated here. If you can, that just helps us improve future webinars. So just a final thank you, everyone, and thank you very much for today's speakers, Laura, Sylviane, and Mark. Have a great day, everyone. Thank you.